Translational control by microRNAs or miRNAs. The regulation of translation is not only by protein factors, but also by small RNA molecules of approximately 22 nucleotides in length that are known as microRNAs or miRNAs. miRNAs form stem loop structures and hybridize to 3' untranslated sequences of mRNA, thus blocking initiation of translation on those mRNA molecules. Since the target mRNA remains intact after miRNA binding, the miRNAs are believed to repress translation rather than stop translation by degrading the mRNA. miRNAs biochemically identical to another small RNA species known as small interfering RNA or siRNA. siRNAs are double stranded RNA molecules of 21 up to 23 nucleotides in length, which mediate the degradation of mRNAs that show perfect complementarity to either of the siRNA strands. The miRNAs are transcribed as primary transcripts, which are then processed to yield pre-miRNAs and form stem loop structures. The pre-miRNAs are further processed by an enzyme called dicer to form mature miRNAs. Dicer also generates siRNAs from its precursors. The mature siRNAs then form an RNA induced silencing complex or RISC that mediates the degradation of mRNAs that are complementary to the siRNA. miRNAs can behave as siRNAs, thus RISC might direct both mRNA degradation and translational silencing. Inhibitors of protein synthesis. There are certain factors which inhibits the translation or protein synthesis. Some well-known inhibitors of prokaryotic translation include streptomycin, erythromycin, tetracycline and chloramphenicol. Inhibitors of eukaryotic translation include cycloheximide and diphtheria and Pseudomonas toxins. Puromycin inhibits both prokaryotic and eukaryotic translation by binding to the A site. Peptidyl transferase attaches the peptide to puromycin and the peptide with puromycin attached at the C terminus is released, prematurely terminating chain growth. Certain antibiotics, for example chloramphenicol, inhibit mitochondrial protein synthesis, but not cytoplasmic protein synthesis, because mitochondrial ribosomes are similar to prokaryotic ribosomes.